Great. So I'm going to talk about some PTSD work that a number of us, big group of us, have been doing in the Million Veteran Program. This work has all been done in conjunction with Joe Galernter and many people at Yale and at other places. Disclosure statement, which I will leave up for about 2 seconds. There we go. Yeah, I want to acknowledge that this research is based on data from the Million Veteran Program. This is a US VA funded program. Just to tell you a little bit about the Million Veteran Program, you've probably heard about it. It's a large biobank, national research program in the US that was launched in 2011 and currently has over 800,000 participants, all US military veterans, who've joined and have provided blood samples for genetic and other data, and have also provided permission for us to link to their electronic health records. And the vast majority have also completed questionnaires. And this is sort of an ongoing recruitment. They actually do hope to pass 1 million veterans in the near future. There's a mental health working group that's led by Joe Galernter, Panos Roussos, and myself. So quickly, you've heard lots about PTSD already. One of the very interesting things about PTSD is that we know when it starts. We know that it has causes. It comes on after specific kinds of trauma. We know a whole lot about the epidemiology of the kinds of exposures that can increase risk for PTSD. What we're obviously interested in today is specific genes for the disorder. And just to be consistent with what you've heard from some of the other speakers, particularly Erica, PTSD symptoms after trauma do occur not necessarily with a normal distribution, but with a distribution. Some people have very few symptoms, some people have more, and we quite arbitrarily draw a line where the disorder is. So I'll come back to that a little later. What we did in this study that I'm going to tell you about was work with the electronic medical record that the VA has. And we've worked both with a binary, diagnostic phenotype, as well as with a continuous phenotype. We spent a lot of time working to develop an algorithm that would give us a good read on diagnostic binary phenotype. You have PTSD or you have not had lifetime PTSD. And that took a lot of work, several years of work led by Kelly Harrington and others in the phenotyping group in MVP. And if anybody's interested, the methods for what we did and what the result was were published in the journal Traumatic Stress last year. Just to jump to some findings. So using this binary case control GWAS for PTSD from the algorithm that we developed, we had 36,000, little over 36,000 European PTSD cases, little over 178,000 controls, and we found 3 genome-wide significant loci that seem to relate to these 3 genes here, one of which, MAD1L1, has come up very frequently in schizophrenia, bipolar, and depression analyses. So that was with the binary case control analysis. But as you've heard already, PTSD is made up of a set of what you can consider symptom subdomains or subphenotypes. This is at least the way that it's identified in DSM-4, where there are these 3 symptom domains. Re-experiencing is obviously very central to PTSD and is what I think differentiates it most from other mood and anxiety disorders. But you also have the hyperarousal domain and the avoidance domain. If we were to have had measurements for DSM-5, we would have had an additional negative cognitions and mood domain. And I'll come back to that later, but I think what DSM-5 does in terms of having that additional domain is it links PTSD even closer to major depression. You'll get a hint of that later. I'm going to show you data that we've already published. This is led by Joe Galernter. But we had available in MVP, in addition to the electronic health record data, questionnaire data. And one of the questionnaires that individuals filled out was the post-traumatic PTSD checklist, civilian version, DSM-4 version that had 17 self-report items, 5 for re-experiencing, 7 avoidance, and 5 hyperarousal. And that's what it looked like in the actual survey. These are the 5 symptoms for re-experiencing. Individuals rate them as having not at all through extremely. These are data that were published in Nature Neuroscience last year, where using this continuous trait, we found a number of genome-wide significant loci that were associated with the trait in the European American sample. What we've done subsequently is to look at all 3 of the subphenotypes, or the 3 symptom domains that make up PTSD and DSM-4. We've also increased the sample size for the re-experiencing sample. And so you can see a number of genome-wide significant loci for each of the domains, some overlap like TCF4, MADL1, but you also see some interesting differences, at least in terms of which of these are genome-wide significant for which subdomain. But interestingly, when we look at genetic correlations between these subdomains, they're actually very high in all directions, suggesting that there may be relatively little in terms of genetic differences or genetic underpinnings of these 3 subphenotypes. We'll come back to that again. So what we have done subsequently is really tried to harness as much information as we could from the phenotype by actually using the total PCL score. So this again is the DSM-4 17-item version. What you see here is among the European Americans, the Manhattan plot, and the closest genes that we see for this continuous trait. Now, when we dig a little deeper and look at the relationships, both at the phenotypic level, which is up in the upper half, the yellow of this graph, and on the other lower diagonal are the genetic correlations, we can see some interesting things. So here we see heritability for, here's for the total PCL, for re-experiencing, avoidance, and hyperarousal are all quite similar. Actually, our algorithmic, which is our binary case control definition of PTSD, is somewhat less heritable here. You can see if you look at the 3 symptom subdomains for avoidance, hyperarousal, and re-experiencing, they're still fairly highly correlated at the phenotypic level between 0.8 and 0.85. But when you look at the genetic correlations among those 3 subdomains, they're even higher. Here is a nice summary that Gita Patak and Renato Palamonti's lab put together for us that summarizes the genome-wide significant findings by Europeans, who are in circles. And we had a few genome-wide significant hits for African Americans, which are in these triangles, for each of the subdomains. So for the PCL total, for the avoidance, for the hyperarousal, for the re-experiencing, and for the case control. So it's a, don't expect anybody to be able to see all the details on the slide, but you can see that we've got some genome-wide significant hits, which are there for case control status, for multiple subdomains. Here's a couple others here, PLEC-HM1, and others which seem to be more isolated. We took a look at the PGC, at their latest case control phenotype, and looked to see whether or not polygenic risk scores from the Million Veteran Program, either using our case control definition or our 17-item PCL total definition, how those worked in terms of mapping onto the PGC PTSD case control phenotype. And as you can see, both versions of our polygenic risk score do a good job of predicting. They explain relatively little of the variance, but they are highly predictive. This is something that we're seeing for pretty much every talk that we've heard today, not even just in the PTSD domain, that there's a lot of genetic correlation between our disorders. So here we've got our case control PTSD definition on the top. Here's our continuous PCL17 total version on the bottom. And you can see a number of significant negative genetic correlations with things like IQ, college completion, which is resonant with what's been seen in the PGC results as well. And then a number of positive genetic correlations with things like neuroticism. Interestingly, we do get some differences in magnitude of genetic correlations between case control and the PCL17 continuous definition. One of those differences seemed to be on depressive symptoms, but overall the pattern of correlation seems to be similar for these two constructs. This is work that Frank Wendt at Yale did. Just briefly, this is using MT-COJO to take the PTSD, this is the PCL17 total results, and to adjust using external, mostly PGC data sets to adjust for the genetic effects of autism, major depression, anorexia, anxiety. This is doing it one by one. And you can see that the overall observed heritability for our PTSD definition starts to decline. And the biggest decline is actually seen with ADHD, which is interesting. And even when you consider all of these disorders together, trying to adjust for their genetic effects, we still wind up with significant heritability for PTSD. This is using genomic SEM, also something that Frank conducted. And so this is a genomic SEM model with confirmatory factor analysis indicating that there are two correlated factors. So one of the factors are our 3 PTSD subdomains, which themselves are all highly related to this one factor. And then over here, we took a look at associations, relationships with anxiety, with major depression, and with neuroticism. And that defines a second factor, which is also related in some ways to PTSD through primarily the hyperarousal dimension of PTSD. This is using a DSM-4 definition. I think it's very likely, although it remains to be actually tested, if we'd had the PTSD DSM-5 negative cognitions and mood dimension here, my assumption would be we would have pulled in a lot more genetic correlation with major depression. But that's something that can be tested empirically with PGC data. And lastly, these are some very preliminary work that we've done to try and get a sense of what drugs might be useful to treat PTSD, and that would be coming from these genomic data. So what we did here is we selected genes that were associated with our PCL total, or PCL17, and used PredictScan to prioritize genes that had predicted expression regulation in at least one of 4 tissues that we'd identified using partitioned heritability as being associated with this trait. And so those 4 tissues were overall cortex, anterior cingulate, frontal cortex, and the nucleus accumbens. And what we did then was we imported these 8 genes. So first of all, just here's the 8 genes. And these are standardized effect sizes, and you can see that some of them are associated with lower expression in PTSD, some are associated with higher expression, and you can see that there's some differentiation among the different tissues. We then went to a number of drug-gene interaction databases to see what this could tell us about drugs that might be useful to influence these effects in these regions. And what we find using these drug databases is that for the gene PLECHM1, there are a number of drugs that have been noted to affect expression of that gene. Here, because we see increased expression associated with PTSD symptoms, we want to knock down the effects. And so drugs that would do that would be dopamine antagonists, alpha-2 adrenergic antagonists, and angiotensin receptor antagonists, all of which either have been quite extensively studied for PTSD or are being studied now. The other drug that comes up, and not so surprising, is CRHR1. Here, it would be predicted that in frontal cortex, an antagonist would be useful in PTSD, but interestingly, it would actually predict the CRHR1 agonist would be useful in nucleus accumbens. And so this differentiation may explain part of the problem that we've not been able to get CRHR1 antagonists to actually work in PTSD. It may be that the story isn't so simple and that there may be effects that are counteracting in different brain regions. So I'm going to stop there. I just want to thank all the many people involved with MVP, and particularly this work. Joel Galernter, Renato Palamonti, Dan Levy, Sam Cheng, Frank Wendt, and Gita Patak, and stop there.
Very good. Well, thank you, Mary. We have a number of questions here. I'd see at least two. Let me just find this again because I had them right in front of me here.
Sure. See if I can find them.
Yep.
Shall I go with the first?
And then,
okay.
Yeah, maybe Chris can read them again because I see them.
So there's the first question. So some PTSD patients have psychotic symptoms and some schizophrenia patients have post-traumatic symptoms. So what do you think about the genetic overlap between PTSD and schizophrenia?
Yeah, so I mean, we do see that. You probably saw that in the genetic correlations from the LD score regression that we do get association with schizophrenia. We had actually hypothesized that there might be biologic relationships between the re-experiencing symptoms in PTSD in general and schizophrenia, things like hallucinations. We have looked in a preliminary way in a smaller sample, we haven't done it yet in MVP, to look to see whether or not individuals who had very high re-experiencing symptoms might have higher polygenic risk scores for schizophrenia, and it doesn't seem to be the case. We haven't yet had big enough samples for individuals with PTSD who actually have psychotic symptoms to be able to test that, but I think that's an interesting hypothesis.
So then the next question is kind of centered around the sample selection bias for MVP, specifically if education is associated with likelihood of being a veteran as an example. Might this generate spurious associations? And I guess maybe not necessarily sample selection bias, but maybe expand on the potential gender bias, male versus female, if this has been studied or will be studied in either of these large scale efforts now.
Yeah, so I mean, MVP is predominantly male. It's about 90% male. And so we're really limited in terms of what we can say about PTSD in women. There are some indications from the PGC and elsewhere, and certainly in epidemiologic data, that women have different risks for PTSD than men. Part of that is difficult to disentangle because the kinds of trauma that women tend to experience, at least outside of combat, are quite different than the kinds of trauma that men experience. So that it's difficult to disentangle, but we're limited with what we can do in MVP. And I think the PGC is likely to be able to do a lot more in terms of looking at sex and gender differences in PTSD.
Yep. And the next question is directed towards different clinical assessment measures. So specifically the correlation you showed re-experiencing case control using the PCL reported measure. I'm not sure if that was PCL in the MVP, but question is, how does this hold when you use other assessment tools like the CAPS 4 or 5, for example?
Yeah, I mean, I don't know. It's a good question. You know, the PCL, as somebody noted earlier, I think is really, at least the version we're using, is asking people about symptoms in the past 30 days. And, you know, how does that relate to, you know, ascertaining a lifetime diagnosis of PTSD? One of the slides, I didn't highlight it, but the genetic correlation between the lifetime diagnosis and the current PCL is extremely high for, you know, whatever reason. I don't know about the CAPS. You know, that there the situation is, you know, you can get lots of data on self-report, as Erica was talking about earlier, from things like the PCL, and considerably less data from the CAPS. We do have subsets of MVP where the CAPS has been administered, and it'd probably be a good idea for us to kind of dig a little deeper there and see what we can learn from those data.
And maybe last question, and then we leave it to the private room, is a comment really interesting, genomic SEM findings. Are these data published, or maybe I can add to this, when or where will they be published if they haven't been published yet?
They haven't been published yet. We have the paper under review, so hopefully soon.